Hey everyone, I am Sergio, a PhD student at the Hashemi Lab in Imperial London, and in this new video tutorial, I am going to guide you on how to use a new feature of a web application that we've just published, the analysis kit, to analyze data from experiments using fast scan control absorption voltammetry, or FSCAV. Now, FSCAV is a technique that is very similar to FSCV, but allow us to monitor tonic changes of neurotransmitters in vivo. If you don't know how FSCAV works, I would suggest looking at some of the literature that I provide below before watching this tutorial or using the application. And so we can start by showing first the homepage of the analysis kit. And then going down into the application sections, we can see that it can be opened just by clicking into the bottom of the FSCAV section that says Open Application. And then after that, a detached window will then pop up showing the FSCAV application. And so in the new window, the first thing we see is a graph on the right that is empty now, and a group of buttons that will allow you to do some actions. The first thing I would like to explain about the application is that it has a fitting and a prediction panel for FSCAV, and they work in different ways. The fitting panel has the purpose of getting a calibration fit, which is commonly obtained from an in vitro calibration of an electrode, while the prediction panel has the purpose of converting your FSCAV acquisitions into concentration predictions based on that calibration fit. Usually the first step is to get the calibration fit, so we will try to explain what the fitting panel does first. And so in the fitting panel, the user can upload FSCAV cyclic voltammograms used to estimate the concentration of the neurotransmitter, as well as the known concentration of the solution used to get those files, and the units of the concentration and the frequency of acquisition of your acquisition board. The structure of the uploaded files have three columns. The first one for the time, then the voltage program, and the current registered for the cyclic voltammogram. And examples of these serotonin FSCAV file can be found in the homepage. Now, as mentioned in the previous video, the acquisition frequency can be found in the specifications of your acquisition board, or simply by calculating the inverse of the sampling period of your CV, which is the time it takes to acquire consecutive samples in your CV. Now, several repeated CVs with the same concentration label, basically taken from the same solution, can be uploaded at the same time, just by selecting all of them. And cyclic voltammograms from different directories can be added consecutively by using the add button every time you change the directory. And once all voltammograms have been uploaded to the application, then the finish button will include all of them and the labels that you have assigned to them into the application. And so there are several fittings that can be used to fit the current data to the concentration labels. These are extensively explained in the publication provided in the video description. And the model type to fit to these current data can be selected in the configuration panel at the top of the window. The simplest one is a linear regression between the charge of the Faraday peak and the concentration labels. When all the signals are uploaded, the application will automatically detect the integration points and the maximum amplitude of the Faraday peak by using local minima and maxima algorithms. Now, these algorithms are designed by default to work on serotonin FSCAV signals, and they can be changed to adapt to dopamine FSCAV signals, which use a different algorithm, just simply because the cyclic voltammograms between these two analytes are pretty different. The parameters used by the algorithms, for example, these selected local minima and maxima, can be also tuned to adapt to a different type of signals. For example, if a different analyte is ever analyzed with this application. And additionally, in case the web application fails to automatically get the integration points, these can be changed manually by setting them to a particular sample across the whole dataset, or for each individual CV by allowing graph selection and moving each point in the graphical interface. Now, when all the cyclable thermograms are labeled appropriately, we can click Fit, and in the second graph, a calibration curve will appear, as well as some goodness of fit metrics. The plot can also be changed in the configuration panel that we showed before to show true versus predicted values instead of the calibration fit. And so once all the CVs are labeled appropriately, with the integration points for the Faraday charge, we can click Fit, and in the second graph, a calibration curve will appear, as well as some goodness of fit metrics. The graph will be different depending on the type of model that you're using. If using neural networks models, a calibration line will not appear because neural networks models do not have a calibration line, but instead, a true versus predicted values will show. And the plot type can also be changed for the particular case of linear regressions in the configuration to show true versus predicted values. So moving on to the artificial neural network calibration models, two of them work very similarly to the linear fit, so we will start from them. The first one is the single electrode model, which is trained with calibration signals from an individual electrode, and this model not only uses the Faraday charge as a linear regression, but also the integration points and the maximum amplitude of the Faraday peak to train the model, and that's why they're included in the graphical user interface. A series of training parameters and neural network parameters can be tuned in the configuration to change the model and fit better for your signals, particularly depending on your dataset, whether your dataset, your calibration data is extensive or not. And once the right model has been selected, just simply by clicking Fit, the application will show us the result of the fitting. It might take a bit longer than the linear fitting, since the neural networks use an iterative process to fit the data and not a deterministic process like a linear regression. And then finally, the pre-trained model works identically. But as explained in the publication, this model has been pre-trained with 140 post-calibrations of trace buffer serotonin from 10 to 100 nanomolar, and with carbon fibers that were cut to 150 microns of size. So if your experimental characteristics are the same as ours, and that's the type of electrodes that you use and the range of serotonin that you're measuring, you can benefit from a large dataset pre-training and then finally tune the neural network to provide a calibration for your particular electrode. So finally, the last model we can select is a neural network that uses the whole cycle thermogram to predict the concentration. There are a couple of comments regarding this particular model. First, the model does not require a post-calibration. That means it's been designed to be an standardized model, to work across electrodes, and it'll be able to get predictions from an electrode without having a specific post-calibration. That means there is no need to upload any fitting signals, and we can directly move on to the prediction panel. And then second, the neural network in this case, it has been trained with post-calibration with 140 electrodes of 150 microns of size, and for serotonin in the range of like 10 to 100 nanomolar. This unfortunately means that the model cannot be used for an experiment with different characteristics because it's been trained from signals from these particular electrodes and for serotonin in that particular range, and it won't work for any other experimental paradigm. Once the model is selected, we can move on to the prediction panel, and here we upload the FSCAV signals that we don't know the concentration of. And we use the loaded model to predict the concentration that is being preloaded in the web application. Now, the upload system in the prediction panel is similar to the one in the fitting panel. In fact, they're almost identical. One important thing is that the software in this case will sort the files by name. So be sure that they're sorted properly in your directory before uploading them. That is because when getting the predictions, the predictions are gonna be dependent on time, and you'll probably want them sorted so they don't mess up in the application. A good way to do this is by giving them a numeric value, or basically their name being a number or the particular time at which it was taken. So once the files are uploaded, the integration points will be found, similar to how it works in the fitting panel. For this particular model, we don't need the integration points, but they're used when you use a different model as the ones we've shown before. And then basically when all the signals are uploaded, we can simply click predict, in which case the predicted concentration will be shown in a scatter graph on the second graph. The calculated charge can also be shown on its own, which might be useful to look at the charge of the Faraday peak trend when no calibration is available for that particular electrode, because the charge trend might give you a relative information regarding the changes in concentration of the neurotransmitter. And finally, both the fitting parameters and the predictions can be downloaded into an Excel spreadsheet. The neural network TensorFlow model can also be exported into a JSON file, both the one that is preloaded and the one that is trained in the web application. And this JSON file can be imported into another TensorFlow platform, such as the one in Python, for it to be used there. And so that's all for this video tutorial. I hope you have enjoyed it. Just a couple of comments left. If you're interested in looking at the code that runs the application, it is freely accessible in the Git repository given in the description of the video. Finally, for any additional comments regarding the application or any reports of bugs or suggestions that you might have, feel free to message me directly by email or contact the Hashemi Lab Twitter. We'll be happy to hear back from you. Thank you very much for watching, and I hope you have a nice day.